Hello, everyone. We'll be starting in more 10 minutes. Thank you for joining. And meanwhile, we begin with today's session. Maybe you can introduce yourself in the chat box and tell us about your educational background and research interest.
Hello, everybody. Good morning. Can everybody hear me?
Yep, we can hear you.
All right. So we will start today's webinar. And good morning, everyone. My name is Jabs, and welcome to this session. I want to start by introducing our company vision and team that stands behind the various resources and training programs that we will go over today. Omics Logic is a program for bioinformatics training that was developed in collaboration between Pine Biotech and Tauber Bioinformatics Research Center at the University of Haifa. Let me just minimize this on the side. Oops. And the company was started as a result of technology transfer from the center, which was founded by Dr. Alfred Tauber and Dr. Leonid Brodsky. Dr. Alfred Tauber comes from a medical background, has extensive expertise in oncology, immunology, and biochemistry of inflammation, and has been recognized internationally for his generous support of many research projects and initiatives that support data science training and applications in medicine. Dr. Leonid Brodsky is a director of the Tauber Bioinformatics Research Center at the University of Haifa and is an expert in bioinformatics and biostatistics. He has numerous publications in mathematics and bioinformatics, and over the course of his career has developed multiple novel algorithmic approaches to biomedical data analysis. The rest of the team includes experts in bioinformatics, computer science, and training. Some of our team members will be presenting today as well. Omics Logic Bioinformatics.
Yeah, I'm sorry to interrupt. I think you didn't press play on the presentation, so we can see your view of the slides.
Let me see. Sorry for that technical. Can you see my screen now?
Yeah, press the play button.
Is that better?
We can still see the same thing. So do you have another screen or something?
No, I don't.
Can you maybe just quickly close it and put it back on, because we can still see, or just close the side. You see the side, the edit screen, just so that we can see the main screen. That's fine.
Can you see my screen now?
No, it's the same thing, but it's okay. Let's just keep going. Just go through the slides manually.
Okay. So Omics Logic Bioinformatics training offers professional certification based on an award-winning curriculum and support of research project. And we have served over 20,000 students around the world and former relationships with universities and networks that can now offer easy access to such trainings in the US, India, Africa, and South America. The goal of Omics Logic programs is to provide training in bioinformatics, enabling independent research guided by mentors and peer example, developing a growing community with shared passion for data-driven research and appreciation of citizen science, enabling students, clinicians, and faculty of all backgrounds to develop novel and independent research using latest technology in life and data sciences. We do this by leading each participant through a well-defined learning process. In order to create a clear path of skill development, Omics Logic employs the Bloom's taxonomy levels of learning. This approach starts with the basics, recalling facts and understanding the terminology, ability to remember the material. With this base, the learning builds to conceptual knowledge, being able to understand concepts and apply information to new situations. Then the goal is to be able to analyze data by drawing connections and evaluate when to use certain methods. This leads to the highest level of learning, the ability to design and create original work. In the program, we will utilize technical aspects of the Omics Logic portal, designed to make this process flexible. Students can learn anywhere and at any time. Their progress is quantified and activity is tracked using a specifically designed point system. After acquiring basic skills, quizzes offer an opportunity to check comprehension. The central focus of this activity is a profile where users acquire levels and badges. Each module will have lessons and assignments that come with an independent certificate of completion. Next, our team will demonstrate the various options so that you are familiar with the process and can make most of the program. Now I'll pass the next presentation to Sonalika.
Thank you so much, Japs, for the introduction of the company and the team. Now, I would like to take you all through the program page and also discuss some of the important things that you all must know by being a part of this particular program. So the very first thing that is important to get started is to have an account on the Omics Logic Learn portal. That's absolutely free. So the link is in the chat box. If you have not signed up yet, please sign up. You can sign up now as well, and if you face any technical issue, then we can assist you. The very next thing, once you sign up, is that you update your profile. So to get started, just click on your email ID that's up here in the top menu bar. That will redirect you to your profile tab. Please make sure that you have an updated profile with the profile picture. Put your name in the title case because you will get an automated certificate once you complete a particular coursework under the program. Please make sure that you have all your social media accounts connected so that we can feature your profile on your progress, and also have an updated bio that will include about your educational background and also your research interests so that our mentors and the community knows about your interests and background. The very first thing to get started is to make sure that you have the starter subscription active as a part of the program, because this starter subscription is going to give you the access to all the starred lessons on the Omics Logic Learn portal as a part of the program. And also please make sure that you have the badge metagenomics in your profile that will give you the access to the program page. Now I'll quickly take you all to the program page and show you what this program page includes and why is it important to keep a track on this program page. So this is the page for metagenomic data analysis, the summer program that we are starting with today. Thank you so much, Jesse, for sharing the link in the chat. Please follow this program page. On this page, you will find the links to the whole session schedule. Now here you can see that you have the calendar on which all dates we have all these sessions scheduled. And also you have the topics that we'll be covering. Along with each and every topic, you have a list of associated online resources. Now, these associated online resources are actually the lessons on the Omics Logic Learn portal that you are required to follow as a part of each and every session. Now, after the session is complete, you can see the recording of the session here on the program page itself. It's important for you to go through the recording once again to revisit the topics that we discussed and also complete each and every lesson as a part of the program. Once you complete or go through the recording and also complete the associated resources, you'll find a complete session icon on the program page under each and every session. You need to click on the complete session icon and do so for each and every session to complete the program. And once you scroll down on the program page itself, you'll find a list of resources that you are supposed to complete. So you need to go through Introduction to Bioinformatics, Bytes and Molecules, Metagenomics, and also the example project of metagenomics. We'll discuss more about all of these resources in the session today to get you all started. And last but not the least, we are going to stay connected via our Skype group. So I'm going to share the link to our Skype group with all our participants, with all our program participants, and you will stay connected with the mentors who, in that group. So whichever questions you have, please post them in the group so that the mentors can assist you. That was it on my end. And now I'm going to pass on the stage to Mr. Ilya Brodsky to get started with today's session. Over to you, Ilya.
Thank you, Sonalika and Japs, for this introduction. Let me share my screen. Hello, everyone, and welcome to this first session of our metagenomics training program. So what we'll do today is I'll talk briefly about some of the basic things that you have to know about metagenomics. For some of you, this will be a review. For some of you, many things might be new, but I'll also point you to some resources on our portal where you can find important information, either on the basic terms that we will use throughout the program, as well as some practical exercises that you can get started with. So like I said, I will talk a little bit about some of the terminology, some of the methods that are used to generate the data. And I'll also talk a little bit about the types of projects that we will review or case studies where you can really see how metagenomics is used. Now, together with myself, Dr. Raghavendran Lakshminarayan is going to be mentoring the program. And in the next session, he will talk to you more about the technical details. But also, this is a program that is designed for you to succeed. And so we're hoping that you will update your profile, let us know about your background, your research interest, or your industry interest, and really allow us to connect with you. Our team will reach out to you if you have any questions, and we have a support group on Skype or on Slack where we can interact and address any questions that you might have. So the purpose of today's session is to discuss the microbiome, how we can use metagenomic sequence analysis to study the microbiome and understand how microorganisms interact in their natural environment, looking at the community composition and structure, thinking about species richness, and understanding the functional roles that some of these microbial communities can play in many areas related to human health and disease, as well as biotechnology and agriculture. So the program is really designed to introduce different types of case studies that illustrate the way the field of metagenomics has been evolving. What are some of the practical tools and skills that you have to be able to analyze this data independently? And also, what are some practical tips, tutorials, and maybe skills that you should have to be able to integrate this quantity of information from different databases, from the reads in the FASTQ files, and also the different metadata that you have about your samples to produce plots that help you interpret the data biologically. So you'll be able to, by the end of this program, process a data set, understand the composition, ask questions about the microbiome, and review literature that will help you find answers and make biological sense of the data that you have analyzed. The way the program is organized is it's really divided up into several different stages. The first one is logic, then we will talk about practice, and then we will kind of transition into a project stage. So why is logic important? Logic is important because we need to understand what kind of data are we going to be dealing with, and what kinds of questions could potentially be asked of this data. Now, different types of data represent different types of biological phenomena. And so we can generate data that describe this biological phenomena, but depending on what kind of data we have about maybe the phenomena, maybe the number of samples, or the way that we plan to process and analyze the data will determine what kinds of questions we can ask. So moving a little bit more into the practical domain, we will talk about different types of analytical approaches. Now, the analytical approaches can be divided into two types. The first one is processing, or really preparing the data for analysis. And then the second one is more on how to work with the processed data and make sense of it. And that can include several different things. So one of them is kind of a standard process, which we call a pipeline, where we take the input files, we go through several different steps, and we get some predefined output. In many cases, that kind of an output requires further investigation. And that is driven both by statistical analysis, visualization, machine learning, and sometimes it's driven by a biological intuition. And so that's where we cannot cover everything that potentially could be done, but this will be more based on a case study. So in each case, we will see what kinds of questions are being asked, what methods are being used to process the data, how is that data then analyzed, visualized, and annotated, and then what kinds of interpretation or conclusions we can arrive helping to study and find relationships between phenotype and what we find in this microbiome or metagenomic data. So this will be covered in several different sessions. Some of the sessions will be live, like today. Some of them will be pre-recorded. Today's session is live. You can ask us questions. You can comment. If you have any specific things that you expect from this program, we would love to know. But in many cases, we want to give you the flexibility of going through the topics that we have planned. And so that is why all of the sessions will be recorded, but also sometimes you will have access to a structured material on our Omics Logic portal, which will include text, images, video, quizzes, exercises with coding or running pipelines. And that's why it's important to get familiar already with the portal, how it works, and how the different content present on that portal can be leveraged. And there you will find some basic coursework, as well as practical exercises in R and Python. We'll primarily talk about R because it's extensively used in metagenomic data analysis, and also because it is providing us with very nice looking figures that we can directly use in research publications. But the online portal really covers the basics, some practical, and gives you some case studies that are structured around publications where experts have been analyzing the data and coming to those conclusions. So it's a good way to understand not only how to technically approach a subject, but how have experts designed, sequenced the data, and made the interpretation of the results. Together with the portal, we will be using the T-bioinfo platform. So the T-bioinfo platform is a point-and-click interface that allows you to analyze big data without having to download the data to your computer, without having to learn how to write code in Bash, and really connect multiple complex analysis methods into a process, a pipeline, where you can get results that are interpretable in a quicker way. So to demonstrate how this is used in the context of metagenomics, I will show you a brief example, and then we will talk about how that can be used either to study metagenomics or to understand the relationship between phenotype and the specific types of data that we will be working with. So let me transition from my slides to the actual portal. Now, if you have already signed up on the Omics Logic portal and you have access now to the program, your account information should be should allow you to log in to this portal, to this platform called the T-bioinfo platform. So I'll pause here. Thank you, Ojasvi, for sharing the link. Please let me know, those that are participating and want to follow along, if you were able to get into this link. And also, if you hover over this profile, if you can see your email address here on top, so you know that you're logged in. And maybe you can share that in the chat. Yes, no, maybe something like that. So I will give you just a couple of minutes, and then we will go on from there. Okay, if you still have not created an account, then I recommend going first to the Learn portal. And on the Learn portal, you can easily create a free account. Unfortunately, if you're just creating your account today, you won't have access to the portal, to the platform right away. It will take a little bit of time for you to receive those credentials. So if you don't have the credentials, you can just observe. But for the next session, I highly recommend to try and log in. And if you're not able to log in or you have some issues with logging in, please please reach out to our support team at support@pine.bio. All right, so let's move on. And here you will see a section, a table called areas of analysis. You will see here many different types of analysis included: transcriptomic data, genomic data, epigenomic data, etc. And right here at the bottom, we have a dedicated section for metagenomic data analysis. So metagenomic data comes in several different formats, and that's why we have them divided into several different sections. We'll talk about some of them, and we'll use some of them quite extensively to analyze data. You will find that the selection of methods is driven in many cases by the type of data that you have. And so we'll talk a little bit about the types of metagenomic data in just a few minutes. But today I will show you a brief example of a specific type of data called amplicon 16S ribosomal RNA gene, where we amplify the 16S ribosomal RNA gene to understand the composition of a sample. And so this link right here will give us access to a demo. In the demo, we can build a pipeline without having to upload data and right away get results, so we don't have to wait for the processing time either. So if you do have access, you can click on there and you can follow along with me. If you don't, you can just observe. And again, you will have an exercise for next time where you can follow along. Now, every time that you click on a button, you get a description that explains what are we going to do and how. And so in this case, we're going to be using the Data2 pipeline. Data2 pipeline was developed specifically because this kind of data is noisy. It has a lot of issues that can make finding signal difficult. And so because it is made to denoise the data, we have to go through a whole series of steps to first clean up the reads and prepare them for the downstream analysis. And so that's what we're going to do. First, we're going to check our quality of the reads. Then we're going to select only reads that are paired. After that, we are going to merge those pairs together. Then we will assign operational taxonomic units to those merged pairs of reads and remove chimeric reads that came from several different locations. So they kind of create this hybrid that cannot be annotated. Once we do this, we have to use a database, a reference that helps us assign each one of those sequences to a taxonomic level. And the taxonomic level basically has a database of sequences, and the closest sequence helps us align the OTUs with the taxonomy. After we have that information, we want to understand what types of sequences do we have. And to do that, we have to create a phylogenetic tree. The phylogenetic tree in this case can be created using this APE package in R, which stands for Analysis of Phylogenetics and Evolution. Once we have that phylogenetic tree, we can then start kind of analyzing and integrating all of that information to perform statistical analysis and study the composition of our sample. And that can be done with a package called phyloseq. Again, we will go through these packages during our sessions, and we can use them in R or we can use them on the platform. But what they do is they integrate the statistical analysis with plotting, and we will see how this plotting helps us understand what kind of data we have. And to the question in the chat, yes, this video is going to be available for everyone to see, but especially important for those that are participating in the full program. And then we're just going to produce some interactive visualization on the platform that will allow us to study the data set that we have. Okay, so for those of you that followed along, your pipeline connects all of these dots together. And what you have in the end are output files that you can download. And they typically are produced from every step that we selected. There will be something from those steps. But also we have here several plots that we can view interactively. So the first important plot is the taxa proportionate abundance bar plot. Again, you will have a whole session dedicated to how this plot is produced and what exactly does it represent. But here briefly, I would like to point your attention to several important things. First of all, when we talk about metagenomic data that represents the microbiome, we're studying a whole community together. And because we have different levels of depth for the different samples that we have, samples here are on the x-axis, we're expecting a lot of variation. And so we want to perform normalization by looking at proportionate abundance and not direct abundance from these samples. So once we have them, we can now see that specific taxa. Again, here we have the full taxonomic classification written out, but as you remember, bacteria is the kingdom, right? So it's not going to change. But then after this, you see that some of these taxonomic levels are going to start to change. And so as we look at our proportions here, we can see this green taxa, which is Bacteroidetes, vary between our samples. And so the obvious question now is, okay, what does that tell us about the samples themselves, right? This is a lot of detail. How do we kind of look at this detail from a high level to understand what kinds of differences do we have? And to do that, and we're going to, again, go through this in detail in one of our sessions, we use a statistical analysis called non-metric dimensionality scaling, which is a plot in 3D that we can look at, and we can see here each one of these dots represents a sample that we just saw. So here we have the samples on the x-axis, and on the y-axis proportionate abundance. Here in our NMDS plot, we have 3 dimensions that are from the non-metric dimensionality scaling, and the dots represent in the individual sample. So you can see that the samples are of 2 types. Chow, which is a standard type of feed used for mice, and HFD, which stands for high-fat diet. And so as we look around, we can notice 2 major things. The first one is that we have an outlier. And the second one is that actually, if we probably remove the outlier, we would be able to see some pretty good separation between our samples, right? If we find some kind of a way to position this plot, we could find a line that could separate the dots. So of course, that is dependent on removing this outlier. And that is really the essence of this type of analysis that we will go through in the full program. This analysis is going to be iterative, which means that you will start somewhere, you will perform some standard tasks, and then you will start asking questions. What do I see? And what does it mean? And if I understand what it means, what are some next steps that I should take to make sure that I'm getting something that I understand, makes sense, but also is reproducible and could tell me something about the biology that I'm trying to study. So this is a very brief example. Again, we will go into much more detail throughout the program, but it illustrates the essence of the type of bioinformatics steps that you would have to go through and understand to be able to work with such microbiome data. And by the way, if you have any questions, please feel free to ask those questions in the chat. So we talked a little bit about this pipeline. We will talk about different methods of statistical analysis and machine learning. And importantly, we will try to reproduce a lot of the plots and try to understand the relationship between the steps that we do, the code that we write, and the outputs that we have. And so as we go throughout the whole program, you will be familiar with how to use different types of packages, different types of methods of analysis, different types of visual plots that you should be expecting, and importantly, how to troubleshoot all of these steps, because in so many cases in bioinformatics, understanding where the problem is guides you to kind of uncovering the issue with either your code or your analysis approach or your data. So let's briefly talk about microorganisms and the microbial community. Now that we've seen all of this information kind of from a high level, what are we trying to study actually? Okay, so the question is the R code used for each step of the pipeline shown available? No, not all of the code that you just saw was in R. Some of the code was written in C, some of it was scripts in Bash, some of it was in R, and some of it was in Python. So it's really a combination of different types of codes that were put together in a single workflow, and that workflow in the end produces these outputs, which are either files or interactive outputs. But the focus of the program will primarily be on how to use a lot of those different standard steps, as well as some coding in R. So the majority of the coding that we'll do will be in R. Hopefully that answers your question. So let's briefly talk about microorganisms and the microbial community. What are we trying to study, and how are we going to be able to study this community, right? Why is it new, and why is it useful? So the connection between disease and microorganisms has been established already in the 1800s. And already towards the end of the 19th century, laboratory methods for growing microorganisms in a lab culture were developed, helping characterize them with staining. These studies provided important information about bacteria function in isolation. However, it was soon apparent that most microorganisms don't live as individuals, but live in complex communities. And today we know that microorganisms, as they are called microbiota, can be very different in different places because they form a stable community. And so one of the efforts of the community that studies the microbiome is to try to characterize both what is healthy, establish a baseline, and how do we measure deviation from that healthy condition. So here you can see that the number of studies has been exponentially increasing from the time that these methods were introduced. And today we have a pretty good sense of these different established communities in the human body, for example. So here you can see some typical members of the microbiota in the mouth, skin, and other parts of the body. And here you can see the number of studies of published research papers on human microbiota. Now, the majority of these studies focus exclusively on the bacteria, but the microbiome actually includes bacteria as well as other members of the community, including the virome, which, as you know, bacteriophages are important members of that bacterial community that regulate the composition, can infect bacteria, bacteria fights them off. So they do form they do influence each other, as well as archaea and other members as well. So when you think of the microbiome, it's really all of the microorganisms, but when we will study the types of analyses that we will perform, we'll focus on one of them. And typically it's going to be the bacteria. Now, in different parts of the body, there are different numbers of bacterial species. And in many cases, you will find an association between the environment that they live in and the community that they form. For example, acidity levels, access to oxygen, and things like that are important factors that actually determine how this community evolves, and is one of the reasons why we cannot just isolate a lot of these individuals to study them in a lab, because we would have to recreate the exact conditions where they grow. And those conditions are in part dependent on the composition of the microbiome. So it's very difficult to extract the microbiome and actually reproduce it in a lab, even though some attempts are being made. So typically, when we think of the microbiome, we think of the gut and the digestion part of how the microbiome functions, but many other communities are also as important. For example, we can talk about the skin, right? So on the skin, surprisingly, even though we wash our skin all the time, we apply different things to our skin, these bacteria form a very stable community. And the reason is because, of course, there are pores, and the bacteria populate the pores as well as live in different cracks between the skin cells. And so as a result, we can see that bacteria, fungi, and viruses form a stable community that starts interacting with the epithelium in our skin. And that is why some of these studies focused on the skin microbiome are are very important for understanding of how to treat diseases and how potentially these bacterial communities could be interrupted. So to study a lot of this, many different international projects have been started, and those include the NIH Human Microbiome Project, the British Gut Project, the American Gut Project, so different kinds of gut projects, Micro Rosetta Initiative. There's even a project for studying the microbiome in space through the NASA consortium called GeneLab, as well as different types of clinical studies involving the microbiome, like the gut microbiome sub-study for cancer prevention, or Alzheimer's gut microbiome project, or the Nurses' Health Study. So a lot of interesting data sets have been reposited in the public domain as a result of these consortia. And we will go through some of these examples in this program, using them as case studies to really understand what kinds of questions are people asking and what kinds of information can we obtain, for example, about the individuals that are being studied, the microbiome effects on human health, as well as many other applications of the microbiome. So let's go now and talk a little bit more about where this data comes from. So we talked about the microbiome. We talked about where we want to get to. How is this data generated? So the way this data is generated is using what's called metagenomic sequencing. And so to understand the process, we'll kind of go through the history of metagenomic sequences and exactly what is being sequenced. And then that will explain a lot of the things in the metagenomic sequence analysis and our ability to annotate and interpret the results that we just saw. So we'll talk a lot here about what's called 16S amplicon sequencing. And it's a technique that was developed because the 16S ribosomal RNA gene is an established gene that has a common function across many different types of bacterial species. And the variation has specific indications for the levels of taxonomy. So the 16S ribosomal RNA sequencing is also low cost, flexible, and it provides us a long enough read that can actually answer a lot of the questions that we have about the microbiome composition. Why is that? The reason is because here you can see the 16S ribosomal RNA gene. So it folds into the structure and has what are called different regions. So these V1 to V9 are the different regions. And the full gene is approximately 1,500 base pairs long, but it also has specific conserved regions. These regions that we see poking out are conserved, and those are between 30 and 100 base pairs long. Now, in between, you have variable regions, and then you have conserved regions. And so the databases that we will use to annotate where these reads came from actually contain those sequences already annotated by the strains of bacteria that they come from. One of those databases is called the Silva database. We'll use that extensively, but also there are others like Green Genes, RDP, and the NCBI National Center for Biotechnology Information also contains that as well. Now, there have been publications that have compared how this is, how these databases annotate the different taxa using the 16S ribosomal RNA gene sequence. And it comes out that two major things are important. Number one, when everything is driven by users, there's a lot of noise because maybe there are some opposite views on which taxa that came from, but also because someone needs to validate and clean up and constantly kind of interact with the data to make sure that it's up to date. And it turns out that Silva is one of those repositories that is most actively supported, reviewed, and validated. And so that's why in a lot of cases, we'll see people use the Silva database to annotate their metagenomic data. Now, how is this taxonomic classification actually performed? So we have here the ribosomal RNA. So the ribosome consists of two parts, the larger part and the smaller, and they are called the subunits. So we have the 30S ribosomal subunit, and the gene that encodes for that subunit is the 16S ribosomal RNA gene. That gene could be amplified. And to amplify, so we extract the DNA from the microbiome, from the actual biological sample, And then we need to amplify some target region because we cannot sequence the full 1500 base pair long sequence, not with Illumina sequencers at least, but other sequencers could do that. But with Illumina sequencing, which is the most reliable and cost-effective in this case, you can see that the primer sequence has to be designed for specific target regions. Now, different combinations of these hypervariable regions are used for the actual sequencing. Sometimes it's just V3, sometimes just V4, sometimes V3 and V4 together. Sometimes you can add another one of these hypervariable regions. It really depends on the level of taxonomy that you would like to use and the ability to amplify and then sequence those reads. What essentially happens is that with this amplicon approach, if the less you amplify, the less of your data is going to be used. So the less information will you be able to expect from your data. A comparison between the full 16S ribosomal RNA gene, for example, sequenced with Nanopore or with PacBio, compared to Illumina, which would only use one of the hypervariable regions, is that you lose about 50% of your data. So you get 50% more information if you sequence the full gene. Obviously, variation in combinations of the hypervariable regions also play a role. But what you can do as a result is actually look at the different levels, right? So the more hypervariable regions you have, the deeper you can go in the taxonomy. So instead of just looking at the species, which obviously bacteria is just one, kingdom, instead of going through the kingdom, bacteria is just the kingdom. You can go all the way to the species if you sequence everything. If you only sequence some, for example, 2, maybe you can only go to order or family level and not any deeper. But essentially then you can look at the body, for example, and analyze the composition, right? So proportions of different types of taxonomies and compare them between individuals, between groups of individuals, or find associations between individuals or microbiota and some phenotype. So this will be the data preparation, the biological data preparation, and this would be the analysis. Now, this is just based on the sequence data. What other types of data can be used for understanding not only what the composition is, but also, importantly, we want to understand the function. So for sequencing, DNA-based approaches like 16S ribosomal RNA or ITS gene sequencing could be used. ITS is just another gene that is used for assigning taxonomy. But also we can use metatranscriptomics or full metagenomics. So full metagenomics, right here, means that we're trying to not only select a gene for taxonomy, but we would like to then assemble everything that we sequence into full genomes. That's a challenge because we don't know all of the genome. So there's not a comprehensive database to say, these are all the possible genomes, let's just find those. We're always expecting to find new genomes. And because they live in a community, we expect to be some drift, genetic drift, a recombination, and other types of interactions that we really cannot predict. And so that makes whole genome metagenomics challenging. Another approach is to try to understand, well, what genes are being expressed? Again, here the problem is we don't know all the genes that could be present, so how do we measure their abundance, right? How do we denoise this data? And so that is why the majority of the studies are still relying on 16S ribosomal RNA. However, more and more whole genome metagenomics is being performed, and even some metatranscriptomics is being done today as well. In addition to that, we can study the proteins and metabolites that are being produced. And that gives us another important insight into not only what is being done in the community, but how is that community interacting with the host. And to do that, ultimately, we would like to integrate all of this information together. And that brings us to systems medicine approach or systems biology approach to be able to understand how all of this information between the types of bacteria, the types of viruses, the types of hosts that they are in, and the different impacts of variables like diet, drugs, and other things can play a role in this complex system. So we won't go all the way into that portion of it, of how to integrate all that data, but we will definitely give you a good sense of how you can get started with metagenomics, and then hopefully that will give you enough tools to continue and study other methods as well that we will also potentially cover in other programs. So let's talk now about some practical examples and case studies that we're going to review in this program. One interesting repository is the American gut sample. So this is a project that was started, first of all, as a research study, but then was crowdfunded and really offered to anyone who would like to participate to be able to understand several different things. First of all, I would like to understand about my own gut sample. What is in it? What are the proportions, and what is the taxonomy? And maybe something is known about these specific strains or species in my body. Another important insight is that I could compare my microbiome compared to other microbiomes of other individuals. And since I know something about those individuals, for example, what kinds of diets they have or what kinds of activity they do throughout the week, I could then understand maybe where I stand in terms of the comparison. So here, this large dot represents this individual, and the little dots represent some of the other members of this project. And here the same way. So here, this project, the data is accessible online. So what we'll do is we'll actually take some of the metadata, and here you can see just a sample comparison that we will do, where we will take individuals that exercise rarely, exercise regularly, and we will try to extract their data using the sample ID and process this data to compare them to each other. And we'll ask the question, does exercise in fact have any effect on the microbiome composition? Another example project that we'll talk about is the skin microbiome. How do we study inflammation? Acne causes inflammation, of course, skin rashes. How can we actually look at the different types of inflammation caused by these microbial communities and just find associations, right? So again, what's important here is that we are dealing with noisy data. A lot of the information that we need is missing. And when we ask these questions, we ask pretty general questions. And then if we want to go beyond the general questions, then we want to actually functionally validate that what we are saying is true. A lot of times we cannot perform those types of experiments on individuals. So what we'll do is we'll look at a mouse study that actually tries to isolate the impact that the microbiome composition plays in the relationship between obesity and anxiety. Because really, today, when you look at the literature that describes microbial communities, you will see that a lot of that is potentially very impactful, but the question is, is that really true, right? So there's a lot of microbiome, theoretically could be involved in almost anything, but how do you know that it's microbiome and not anything else? So in this study, the authors actually designed an experiment where they took mice just after their microbiome was established. Those mice were fed with different with different diets. And once those microbial communities were established in the guts of these mice, they were studied, analyzed, of course, but then also that microbiome was implanted into other mice, recipient mice. Now, the recipient mice did not have a difference in diet, but they got the microbiome from different types of donors. And because nothing else was different between these mice and just the implanted microbiome, they could test the difference in their activity and in their response to different stimuli, and identify that, in fact, only the microbiome had a contribution to the levels of anxiety, which is a very interesting finding, and a finding that you will find being reproduced in other studies as well. And so where does that leave us? Where does that bring us to? Well, it actually helps us understand many different things about the microbiome. First of all, we know that your healthy microbiome is established in the early stages of your life. And that is primarily driven by the maternal microbiome and your genetics and environment. And as the infant, about 4 years old, that microbiome is now established, but is continuing to be affected by the number of antibiotics that are being used, as well as diet and exercise. And then throughout the stages of that microbiome developing, many different scenarios could be explored. And for many of these, we will look at examples and case studies that really allow us to study these things in detail. And so one of those, we will look at microbial colonization in infants, and how different bacteria get established, for example, between vaginal birth and C-section, so different types of births, as well as different types of use of antibiotics. We will also look at the enterotypes, right? How these different types of microbiota established in the early infant stages are then affected by different types of lifestyles and environmental factors, and ultimately, what are some of the general kind of types of microbiota that we expect? And they basically, for the adult microbiome, are typically separated into 3 different enterotypes that are based and highly associated with both location and diet. And so because of all of these studies today, as you probably all know, you can measure your microbial composition at the preclinical stage, right? So there's a lot of consumer directed kind of tests that you can take, and you know what is in your gut. The problem is that, of course, there's a lot of noise, and I think there's a developing trust into how these applications really work, but it's definitely an exciting area that a lot of there's a lot of interest at the preclinical stage. What about the clinical stage? Well, in the clinical stage, we also are starting to see a lot of innovative uses of the findings coming from this type of research to address very important types of challenges that previously have not been addressed using the microbiome. For example, China approved an Alzheimer's treatment that targets the microbiome. Or there are many studies that look at the association between our ability to metabolize different treatments and the types of responses to drugs. And even being able to develop a microbial signature that will help us predict, for example, in the case of immunotherapy, predict the important toxic responses to immunotherapy that some individuals have. So when we think about the microbiome, this also has a role to play in the current pandemic. And so a lot of people are turning to studying the microbiome to understand how does the microbiome affect the risk of being infected, the length of the disease, and the speed of the recovery. Microbiome is not only used for human health, it is also extensively used in other applications, including biotechnology and agriculture. And we will also look at some example data sets that can help us understand the way that the microbiome potentially could be used as a fertilizer, because it plays a role of kind of, helping, for example, in our body, helping us digest some of the food in the soil, helping the plant roots extract the correct nutrients from the ground, and might be a very effective alternative to the types of elements, chemical elements that are used to kind of perform the same function. So in conclusion, what is this program going to be about? This program is going to help us understand different types of methods of analysis that are used to analyze this kind of data and develop an understanding of the microbiome. As we saw in today's example, we will go through many different types of analyses approaches and try to understand the logic behind these approaches and the types of outputs that they produce. We will then look at some coding exercises from very simple to more advanced and try to understand how to work with this process structured data extract the right kinds of information and kind of look at it as a survey. And how do we analyze a survey? We try to look at the number of people that answer certain questions in a certain way. So we will take the similar approach to studying the microbiome. What I want to leave you with today is a little bit of further reading. So before you continue with the program, and before you start thinking about what should I analyze in terms of microbiome, take a look at these 3 publications. So the first publication is going to be based on this original project, the Human Microbiome Project, and will really tell you about the different approaches, specifically bioinformatics approaches, that were designed to study the human microbiome data. We will also look at the different concepts, strategies, and advances in the microbiome. I'm going to share these links in just a second. And finally, the website for the Human Microbiome Project itself, where you will see both the history, the current status, and future directions for this important project. So let me just quickly share those links with you. And I'll paste them in the chat. All right, so excuse me. At this stage, I really want to open the stage for discussion. Sorry, yes, I might have pasted this. Oh, okay. For discussion. So if you do have any questions, please feel free to ask. And I'll give it just a couple more minutes. So if you have any questions, you can put them in the chat and I'll address them one by one. And in addition to that, I would like to pass the stage back over to Sonalika to talk a little bit more about making sure that your profile is set up, that you know how to access the different times that we will have our sessions scheduled on. And then once those sessions are over, how to access the video material and how to interact with our team using different channels for discussion.
Thank you so much, Ilya, for such a wonderful session and taking all the participants to the resources that we're going to take up in the program. Yes, so as Ilya mentioned, I would now like to assist our program participants with any of their technical issues, if there are any. So first of all, please tell me if you have signed up on the portal. Please put a yes or 1 maybe in the chat box to make sure that yes, you have signed up. I'll be awaiting the responses in the chat. All right. And those who have signed up, have you updated your profile as well? Somnath and Aparna, you can sign up right away. And if you face any technical issues, then we can help you out with that. Swarnali, perfect. Those who have not updated their profile, they can update it as well. And just to make sure, yes, Somnath, I'll just give you the link. All right, yeah. So I wanted to make sure one thing here. You will be signing up on the portal and create credentials for yourself. So the credentials that you create for yourself on the portal, you have to use the same credentials every time you log in to the portal. But for the T-Bioinfo server, that is the hands-on analysis server that Ilya also showed. For the very same server, I'm going to send you out the credentials. Every program participant is going to have an educational license for 1 month. That is the duration of the program. So please make sure that you use the very same credentials when you have to sign up or log in to the portal and the server to run your analysis. I'll repeat once again that you will be creating the credentials for yourself. Please use the same credentials to log into the portal, Omics Logic Learn portal. And the credentials that I'll send you over, please use them to log in to the bioinfo server. Thank you, Ilya, for taking up that question. And now, quickly, I would like to take all the interested participants who are present here through the program registration. So there were many of you were interested to just have a look at how this online training program is going to work. So I hope you all had a good idea today about how we're going to have these sessions conducted and how you will be going through these associated resources, run your pipelines, and also go through the quiz and do the analysis. So now that you are well aware of how this program is going to work, let me just take you through the registration steps so that you can register and get access to all the resources that we discussed today and also become the part of our upcoming sessions. So here we have 3 different subscription levels. On the program page itself, those who have already enrolled, you are not going to see these 3 subscription levels because yes, you have already picked it up. The ones who are interested to enroll, on the program page, you will find 3 levels mentioned as beginner, intermediate, and advanced. In the beginner level, you will get the access to such sessions that we have had. All the resources in all these 3 different types of subscription levels are going to remain the same, which includes such live sessions or recorded sessions, associated resources on the Omics Logic Learn portal, access to all the course material on the Omics Logic Learn portal, and also an educational license on the bioinfo server to run the analysis. The difference lies in the duration and also the access to one-on-one mentorship guidance. So in the beginner level, you will get the access to mentor guidance in such group sessions as we have today. In the intermediate subscription level, you will get a chance to also schedule one-on-one meeting with the mentor. That could be between you and your mentor, and you discuss your questions, your doubts, your ideas with the mentor. And in the advanced subscription level, you'll get some extra days to work on a research proposal. All right, so in addition to the duration of the program, you'll get some extra time period where you work on your research proposal and get started with your independent research project as an outcome of the training program. So that is the main difference between beginner, intermediate, and advanced subscription level. I hope I was able to clearly specify the difference. If there is still any doubt or question, please put it in the chat box so that we can clarify it. All right, so if we have no more questions at the moment, yes, Saranya, you can definitely pick a beginner level first and then upgrade it to an advanced level. Yes, you can upgrade your level anytime in the application. Yes, and the last thing that I wanted to show all of you was this Skype group. The participants who have joined the program already, I'm going to send you the link to join our Skype group. Please make sure that you join this group. The mentors are also here in the group, and you can anytime put your question when you're running a pipeline or if you have any technical issue, just to ensure that your issue is answered on time. Please make sure that you join the channel. Okay, so Ilya Unzail is asking, are there any methods for combining or comparing 16S rDNA and whole metagenome shotgun DNA analysis results? I think Ilya has left. It would be great if he could answer your question, Unzail. So I'm going to reply you back on your question. Please put your mail ID in the chat for me. All right, are there any more questions? If not, then I think we are good to end today's session. Before I do that, I would just drop in our mail IDs in the chat so that you can reach out to us with any of your queries later. You can reach out to us at marketing@omicslogic.com for getting yourself enrolled, and Ms. Parshal will help you get enrolled in the program. Yes, your subscription will be active for the entire length of the program, that is 1 month. And the starter subscription will give you the access to all the start lessons under the courses tab on the portal. Yes, definitely, Harsh. All these options have a certification. So there are two things here. You are going to complete some of the courses as a part of this online training program. You'll get individual certificates for each and every course that you complete. That is point number 1. And also you will get the certificate for your program on successful completion, right? So let's say if I complete my complete metagenomics course and also the example project that we discussed about. So I'll get certificates for both of these. And then when I complete the program, so I get a certificate for my program as well. This is how you can see your certificate. This is how you can see the certificates for your courses, and then for your programs. This is a training program. Please don't get confused between a program and the courses. Courses are available on the portal that you need to take up, and the programs are online meta-guided training programs. There are different training programs that we hold every month, and this is one of them. And for the program, you'll have another certificate. Yes, Unzail, we'll reach out to you. Thank you for sharing your email ID. Thank you, everyone, for joining today's session. And we'll now meet for the next session on May 27th. The session on May 27th is going to be a live session that will be taken up by Dr. Raghavendran. We'll see you in our next session, and the participants will get the program participants will get the invitation link. And also, in case the email drops in the spam box or the promotions box, please drop it or drag and drop it to the inbox, main inbox. And another way to get the access to the session is definitely the program page. Once you're a part of the program page, you will be able to look at the Zoom recordings as well for the Zoom session link as well for the session. So that was it for today's session. Thank you everyone for joining. It was nice to have you all